Okay, and our next speaker, who you can see on our screen, is Justin Young, who is a student from University of Wisconsin-Madison, and he will tell us about identifying spontaneously emerging autocatalytic systems using the liquid chromatography and mass spectrometry. Justin, the floor is yours.
Thank you. Give me a second to fiddle around with PowerPoint.
Okay,
you all can
we can
see
see the slides. Yes, it's perfect.
And I shall begin. Hello, as you guys all may know, my name is Justin, and today I will be talking about the use and analysis of untargeted tandem LC-MS for understanding the origins of life. So the central question is, how did lifelike chemical systems arise? We know from prior research that life processes arose from a heterogeneous, messy mixture consisting of a myriad of different chemicals, with that timeline progressing into some unknown events happening in the middle, but getting ultimately the end product that is life. And so we are mainly concerned regarding what happens in the middle. And one thing that could arise from what happened in the middle is a focus on autocatalysis. Now, an autocatalytic system is a system where a series of entities and certain molecules catalyze their own production, as depicted in the figure on the right, where A combined with a food source produce C, which also combined with a food source produce B, which produces two A's, and so on and so forth. In a sense, this process becomes self-sustaining, but necessitates a food set to supply the initial process. And so the question arises from this is, how do we select for autocatalytic cycle? And to do that, we utilize chemical ecosystems aboard using systems chemistry, which provides a bottom-up construction and a focus on more towards the overall network of chemicals and their respective properties. The framework for attempting to select a chemical ecosystem can be shown in the figure here, where you take a chemical mixture or a chemical soup and transfers parts of it into a new fresh mixture of the same chemical composition, letting that incubate for a period of time before taking part of that new incubated mixture and transferring it into a similarly new chemical tube, and so on and so forth. That one process of transfer and incubation we define as a generation. And to see how the effect of this lineage and history, we compare it with each time with a fresh mixture without any transfers or prior history that's also incubated at the same time period. This basic framework leads over to the experimental procedure, for which we have a well plate consisting of 96 different wells with 24 wells used for the controls, that are just fresh soup made and produced every single time, while another 24 are ones that have this transfer history, where we have the soup chemical mixture, letting that incubate, and then taking 20% of that and transferring it over to the next generation, and letting that incubate and letting that process go on, that cycle of a generation go on for 70 different generations before ultimately analyzing for using LC-MS. And here pictured is the chemical composition of the prebiotic soup, as well as it includes apatite and iron pyrite materials. To analyze the contents of each of the replicates of the wells, we utilize LC-MS. LC or liquid chromatography is used to separate the compounds and obtain the retention times, and MS, the mass spectrometry, is utilized to vaporize and ionize and to identify ultimately the mass charge ratio and fragmentation patterns. We then utilize a myriad of different databases to identify compounds based off these three recorded values, and as well as for the area under the curve for the retention time. That area is used as the area for that describes the abundance of each identified compound discovered. And so what we end up is having the data structure that looks like this after cleaning and making it look nicer, where we have column of the name, the identified name, and formula, if possible, along with the mass charge ratio, retention times, and every respective generation replicates with the values of each cell representing the abundance for that specific compound in that specific replicate. And so ultimately, when conducting our preliminary analysis, we have a series of 3 different goals using this pseudo graph to describe, with the blues representing the controls or those that have no transfers, and reds representing the experimentals or those that have transfer history. The first is to analyze the sample mean at any given generation between the controls and experimentals. The second is looking at the overall population mean of the experimentals and see how that changes over different generations. And lastly was to compare variance between the controls and experimentals and see if there's any difference in variability. And so when looking at the overall picture, the macro of the data, we observed that there's no observable statistically significant difference between those with transfers and those without transfers. This plots the average area for the means of all the compounds, meant to represent what the average abundance would be for any given compound, and from the graph that there is little to no separation between the dots, the control dots, and the experimental dots. Similarly, because the nature of our data has a lot of high dimensionality, or lots of different columns, we conducted PCA analysis for our 3 different data sets that we, for the most part, mainly focused on, those being generations 1, 5, 10, 15, and 45 on the left, generations 34 and 36 in the middle, and generations 67 and 68 on the right. And from these graphs, there is very little separation between the points of the controls and the experimentals. While the case is for the overall picture that there is no significant difference, we then shifted towards a more bottom down approach, looking individually from the bottom, both in terms of generation and individual generations, as well as compound by compound. The first by compound is on the graph on the left. That graphs the mean area for the top 100 compounds sorted by average area for generation 35. While the two lines of the graphs are almost overlaid on top of each other, we do notice some gaps and holes between these lines, not just specifically mainly here, but also not just there, some out up in front there, as well as minute ones towards the end of the rank. Additionally, when doing dimensionality reduction and PCA analysis, we notice that the points of the experimentals are clustered together for the experimentals, and thus we proceeded to do an extensive study done around the surrounding generations, mainly that of 34 and 36, around generation 35. The next thing we conducted was doing the compound by compound analysis for 34 and 36. And what we did is a two-sided t-test for every single identified compound. And what we found was a list of 24 different compounds that were statistically significant in both the generations, 34 and 36, at the alpha level of 0.01. And this is actually higher than what we'd expect at that level, with 24 out of about a little more than 10 times higher than what we expected. Now, the coloring of this table indicates whether the controls or experimentals were higher in a given generation. And so the blue represents that the mean was lower for the experimentals in 34 and 36, whereas the red represents the vice versa, that the experimentals were higher. Now, what I'm saying is that all 24 of these compounds were significant in the same direction. That means if the controls were higher in one generation, that means it's also higher in the other generation, 36, which is something we did not expect. We expected about half to flip direction, but in fact all of them stayed the same. Additionally, when graphed with the value in generation 35, we notice some oscillations between the overall means observed from 34 to 36. These oscillations are twofold. One, that the total mean value goes down and back up and down, but also, as in the case of the graph on the left, but also that there's a flip-flop between directionality, as seen on the graph on the right, with the experimentals being lower than controls in 34, then higher in 35, before becoming lower again in 36. And this may be potentially indicative of one half or one cycle or a transitional period in an autocatalytic system between production and consumption. And these similar oscillations are found in prior experiments, specifically one conducted in 2019, where with orthophosphate and the change in its concentration over a generation of about 12 or 12. Now, when producing the same analysis for generations, for two adjacent generations, neighbor generations, in this case the last two, 69 and 70, what we notice is a complete absence in the same directionality. As by the different colors. And that means that if the experimentals were lower in one generation, it flips to become higher in the other generation. So ultimately, at the conclusion of our preliminary analysis, we came with 4 different conclusions. One is that there's little or no overall difference between controls and experimentals. 2, that there are more statistically significant compounds between generations than what we would expect, even in the previous slide that of generation 69 and 70. The 7 that were identified out of the total of 13,000 was about 5 times bigger than expected value alpha 0.01. There are some signs of lineages and shared characteristics in history by the same directionality found in 34 and 36. And lastly, that the pattern dissipates between 2 adjacent generations, and these possible oscillations indicative of potential autocatalysis and some interesting nonlinear chemistry. And lastly, I would like to acknowledge these people and the members of the Baum and Yin Labs. Thank you.
Thank you so much for your very interesting talk. We are waiting for some questions from the chat, and I'd like to ask you. I could miss it from your presentation, but I'm really curious to hear more about it. How do you decide on the composition of the enriched probiotic soup you're starting from?
Yeah, so the choice of the soup was based on a bunch of different what a bunch of different soups that are previously published and done experimentals on. These compounds that were previously published. That the soup is enriched and it's indicative of what chemicals may be present on early life on Earth under those conditions.
Okay, so you base on previous publications, and I guess it is mainly amino acids and like different, not very complicated, but quite complex compounds. Okay, sounds interesting. Okay, we still encourage you to ask any questions you may have on the chat. And if there is no more questions, thank you so much, Justin, for your presentation. It was really nice to hear your talk.
Yep.
So thank you.